Today, we're going to be going over genetic engineering, all the ways that we can influence genetic variation in organisms. But first, I want you to sign in for daily attendance. And if you haven't already done so, please review the slides before watching the video. I've attached them. And next, I want you to click View on the PowerPoint and show speaker's notes. I have a lot of important notes that will correspond to the slides. Let's start. So how do we increase genetic variation? We introduce mutations, or we can use biotechnology. We're able to change the genetic makeup of an organism. A simple example of biotechnology would be treating bacteria with radiation to induce these mutations. But if we want to dive into something more complex, let's talk about joining together DNA. Remember, DNA is like the hard drive of a cell, an organism. It has all the data needed for life, but it's possible to change this DNA. In doing so, we can create recombinant DNA, which is the joining of DNA from two or more sources, two or more organisms. In organisms where we change their DNA, we call them transgenic organisms, right? DNA contains genes, and these transgenic organisms contain genes from other species. This is completed by recombinant DNA, inserting the DNA and genes into the host organism. And they're known as a GMO, a genetically modified organism. So an example of this would be, we have Pacific Ocean jellyfish. They're fluorescent under certain light. They glow. Their DNA can be taken out and processed and then injected into mouse embryos, which are fertilized eggs which have undergone division, and then the mice will be able to glow under certain light, something that is clearly not natural in mice. But how can we apply recombinant DNA to something important like medicine? Well, we can use it to make substances for medicinal use, for medicine. We can use recombinant DNA processes to make insulin for diabetics, right? Insulin is really important for keeping your blood sugar stable. Some diabetics lack insulin. Their body doesn't produce enough or at all. So we're able to make it so they can inject it, or they might have a insulin pump. Also, we can use recombinant DNA to produce human growth hormone. So simply put, human growth hormone stimulates growth. Some people don't have enough. And lastly, we talked about hemophilia, right? Hemophilia is a disease where you lack clotting factors, right? So instead of your blood being able to clot to stop bleeding and eventually creating, a scab, people with hemophilia will bleed out. But we can create these clotting factors for them so they can live a normal life. Let's see how we can use recombinant DNA in bacteria. So let's start by looking at our picture of the bacteria. Bacteria have some circular bacterial DNA, but many also have plasmid DNA, circular DNA. It's separate from the bacteria's DNA, and it can replicate and express proteins separately. So we can use these plasmids to our advantage. We can use restriction enzymes to make cuts in both plasmid and desired gene that we want to insert into the plasmid to express. So when we insert a new gene into the plasmid, it will be expressed. So for example, insulin, as we're going to see here, right? So humans, in our DNA, we have an insulin gene. But for some people, it's deficient. So we take a healthy insulin gene and we take a plasmid from a bacteria and we combine them together to make a recombinant plasmid, recombinant DNA. And now, just like a transgenic mouse, we have transgenic bacteria. We grow these in a lab and we can extract insulin that can be used for diabetics, people with diabetes. So let's stop and check. Let's answer this question. The accompanying diagram illustrates some key steps of a procedure in one area of biotechnology. The letter X most likely represents A. Bacteria cells that are unable to synthesize insulin. B. Human cells that are able to synthesize antibodies. C. Bacteria cells that are able to synthesize insulin. Or D. Human cells that are unable to resist antibiotics. So right, the answer would be C. Bacteria cells that are able to synthesize insulin. We put the human gene for insulin in a bacterial plasmid DNA. It's inserted back into the bacterial cell. The bacteria cells divide in culture in the lab, and we can extract insulin from those bacteria cells that produce insulin, human insulin. Now let's move on to cloning. I'm sure we've all heard about this. A clone is just a genetically identical offspring produced from a single cell. So asexual reproduction can lead to clones, but some animals who rely on sexual reproduction have been cloned. An example of this is Dolly the sheep. Dolly the sheep, she was cloned in 1996 and lived until 2003. She was a female domestic sheep, the first mammal cloned, and she was created from a female somatic or body cell, not reproductive cell, using a process called nuclear transfer. In the process of nuclear transfer, let's follow the flow chart. So, a donor cell is taken from a sheep's udder, from a sheep's body. It's a somatic cell. Another sheep has an egg cell taken, but the nucleus of the egg cell is removed and the donor cell from the udder is fused with the egg cell. So then the fused egg cell begins dividing normally, divides into an embryo, right? And then the embryo is placed into the uterus of a third sheep, a foster mother. The embryo develops normally into a lamb, and in this case, it was Dolly. And Dolly would have the same genetic makeup as the sheep from where the udder cell was taken, the body cell, not the egg cell, because the nucleus, the genetic information, was taken out. Now let's stop and check and answer this question. The diagram below represents the process used in 1996 to clone the first mammal, a sheep named Dolly. We just talked about her. Look at the flowchart. Which statement concerning Dolly is correct? Choice A, gametes from sheep A and sheep B were united to produce Dolly. Choice B, the chromosome makeup of Dolly is identical to that of sheep A. Choice C, both Dolly and sheep C have identical DNA. Or choice D, Dolly contains genes from sheep B and C. The correct answer would be the chromosome makeup of Dolly is identical to sheep A. When the body cell is taken, while it's combined with the egg cell, only the body cell's genetic material is retained. So Dolly would be a clone of sheep A. She would not be related to sheep B genetically, and sheep C is only like a foster mother, only carried Dolly during pregnancy, but has no genetic relation to her. Now, how can genetic engineering be applied to forensics? So remember, I said DNA is like your fingerprint. So we can use DNA fingerprinting almost. In a way, it can be described as fingerprinting. It's used to identify individuals since DNA varies from person to person. It can be used to solve crimes, but interestingly enough, has also been used in freeing wrongfully imprisoned people, where DNA technology has come out after they were convicted and has proven that they were not at the crime scene. In 1986, DNA was first used in a criminal trial. Let's take a look at this sample right here. The sample electrophoresis gel. We have our crime scene, right? DNA that was left at a crime scene. And we have 3 imaginary suspects, we'll call them. Now, when we put their DNA in the gel and try to match them to the DNA in the crime scene, look who matches. Remember, we said base pairs travel through the gel based on size. So we have a match. Suspect 2 appears to have been at the crime scene. And we're going to wrap it up with some concerns with genetic engineering. So there are some privacy issues. Now that DNA testing and genetic engineering is becoming so widespread, there's kind of an issue. Who owns your DNA? You'd think you own your DNA, but that's still an issue. And what about safety issues? Are genetically modified crops safe? What are the risks? Are there any risks? Remember, we were introduced to golden rice on Wednesday's video. Golden rice was produced from genetic engineering, and it has more vitamins, so people in impoverished countries can be healthier. And lastly, ethical issues. Just because we can do something, does that mean we should? Do we have the right to meddle in nature? Should we? Who gets to decide this? Now, let's end this lesson with this question. Scientists have cloned sheep, but have not yet cloned a human. The best explanation for this situation is that A, the technology to clone humans has not been explored. B, human reproduction is very different from that of other mammals. C, there are many ethical problems involved in cloning humans. Or D, cloning humans would take too long. And remember, this is one of those questions where you should pick the best explanation. So yes, we just talked about the ethics of genetic engineering. The choice would be C. There are many ethical problems involved in cloning humans. It's really an ethical dilemma that so far we really don't have an answer for, and it's not really accepted to study cloning of humans. This concludes the lesson for today. Thank you.